With this video, we're getting close to the end of our first series of videos on pathology. We're going to talk about cellular accumulation, and therefore all the conditions where there is an accumulation of different elements in the cells. These accumulations may be pathological or physiological. But let's start with lysosomal storage. Lysosomes normally have a number of functions in the cell. They originate from the Golgi vesicles that, by becoming autonomous, select specific sets, which will then be activated by an acid pH. Lysosomes are very important for autophagy, a phenomenon occurring to remove any damaged cellular organelle. This phenomenon is firstly mediated by the formation of the double coating on the target organelle carried out by the endoplasmatic reticulum, and then by the fusion of this vesicle with the lysosome. Lysosomal enzymes destroy the inner layer of the membrane, leading to organelle destruction. The residue is a pigment granule, also known as lipofuscin. Autophagy phenomena mediated by lysosomes are extremely important, not only in the defective organelle removal, but also for cell differentiation phenomena. In fact, as we have already seen, some cell structure removal may lead to differentiation, to cell death, and may lead to necrosis. Residues of the lysosomal actions are residual bodies, which are lysosomes with indigested residues inside, lipofuscins, which are residues of lipid peroxidation, and, finally, pigments like carbon and tattoo ink, which cannot be eliminated easily. The function of lysosomes is extremely important, so a lysosomal storage-related pathology may undermine not only molecule digestion inside the cell, but also more important functions as we've just seen. Lysosomal storage diseases may be linked to genes, as for example, the lack of lysosomal enzyme synthesis. Remember that the Golgi apparatus starts its function because lysosomal enzymes are marked with mannose. These lysosomal enzymes gather in a specific point that then buds and becomes lysosome. This is due to the lack of proteins activating the lysosomal enzymes, or due to the lack of proteins activating the substrata of these enzymes. Another example of lysosomal disease is that of gangliosidosis or Tay-Sachs disease, a deficiency of the beta-hexosaminidase A enzyme, which is normally responsible for cutting gangliosides GN2 in the brain. There are three main forms of Tay-Sachs: infantile onset, juvenile onset, and adult onset forms. Another form is that of mucopolysaccharidosis, which is not exactly a disease, but rather 9 different pathologies, different from each other, and each linked to an enzyme deficiency that is responsible for cutting mucopolysaccharides. These pathologies manifest with systematic symptoms, dysostosis multiplex, developmental delays, hepatosplenomegaly, and so on. Then there's glucocerebrosidasis or Gaucher's disease, which is a glucocerebrosidase deficiency. Glucocerebrosidase is an enzyme in macrophages' lysosomes that cuts glucocerebroses and converts them into glucose and ceramide, preventing its accumulation. When this enzyme is lacking, organs like the spleen, liver, and bone marrow fill up with cells that cannot catabolize glucocerebrosides. There are 3 possible situations. Type 1 is the most common form. It is the non-neuropathic disease, characterized by osteoporosis as a result of cells swamped with glucocerebrosides in bone marrow, anemia, thrombocytopenia, and neutropenia. This is due to the fact that the bone marrow is swamped with those cells and cannot produce normal blood cells. In fact, this compression may lead to bone infarction. Type 2 is a pathology also known as neutropenia in childhood or Gaucher's disease, which manifests from the 3rd month of life and leads to early death by the 2nd year of life. This is because besides hepatosplenomegaly, there is also brain damage, ocular movement disorder caused by the accumulation of pathological macrophages. Type 3 is a chronic form similar to Type 2 in terms of symptoms, but with a slower and deferred clinical manifestation.
Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.